All right, today we're jumping into one of the immune system's most awesome weapons, the classical complement pathway. Now, forget about brute force. Think of this as a precision-guided missile, where our own antibodies act like laser sights, painting a target for total destruction.
So the big question we're tackling is this. How does your body take a simple antibody that's just latched onto a pathogen and turn it into a direct signal to unleash a powerful, cell-destroying weapon? Let's dive in and find out. So, we're focusing on the classical pathway. This is a really crucial part of our adaptive immunity. You can think of it as the bridge that connects a very specific antibody response directly to the raw destructive power of the complement system.
The most important point is right here in the definition. The classical pathway is an antibody-dependent cascade. That means it doesn't just fire off randomly. Oh no, it's specifically triggered by an antigen-antibody complex, which makes it a highly targeted attack system. Okay, let's start right at the beginning. Initiation. This whole powerful cascade, which ends in a cell literally being blown apart, all starts with a single, very specific recognition event. It's all about pulling the trigger. But here's the thing, not just any antibody can do this. So a key question, especially for your exams, is which antibodies can actually pull the trigger and get this whole process started? Now, this slide shows a really clear hierarchy. IgM is by far the most potent activator, all thanks to its 5-armed pentameric structure. With IgG, the subclasses really matter. IgG3 is the best, followed by 1, and then 2. And this is crucial. Remember this one. IgG4, IgA, IgD, and IgE, they can't activate this pathway at all. And this explains exactly why IgM is so powerful. Because it's a big pentamer with 5 Fc regions all clustered together, just one single IgM molecule is enough to kick things off. IgG, on the other hand, is a monomer. It's all on its own. So you actually need at least 2 IgG molecules to be sitting close together on the pathogen surface to get the cascade going. The very first protein to arrive on the scene is the C1 complex. It's made of 3 parts, C1q, C1r, and C1s. Think of C1q as a kind of grappling hook. Its little globular heads bind to the antibody's Fc portion, that's the tail end. Now, for a stable connection, you need at least 2 of those heads to attach. And this binding, which needs calcium ions to work, causes a shape change that activates C1r, which then immediately activates C1s. Just like that, the fuse is lit. Okay, now we move into the amplification stage, forming the convertases. You should really think of these as molecular engines. Their whole job is to just churn out thousands of active components, which dramatically ramps up the immune response. Building the first engine, the C3 convertase, happens in a really clear sequence. First, that active C1s enzyme we just talked about grabs a C4 protein and cleaves it into C4a and C4b. The bigger C4b piece sticks directly onto the target cell surface. Next, C1s does the same thing to a C2 protein, and this step needs magnesium ions. The resulting C2a piece then binds to the C4b that's already on the surface, and boom, you formed the C4b2a complex. And here is a super critical point to memorize. The classical pathway's C3 convertase is C4b2a. Its one and only job is to be an enzyme that goes out and cleaves as many C3 molecules as it can find. This creates massive amplification. So when C4b2a cleaves C3, it creates 2 really important fragments. You get C3a, which is an anaphylactotoxin. That means it promotes inflammation. Then you get C3b, which is the real workhorse. It can act as an opsonin to tag cells for destruction, but its main job right here is to help build the next enzyme in the cascade. Now, building the second engine, the C5 convertase, is incredibly efficient. The system doesn't start over. A C3b molecule, one that was just created by the C3 convertase, simply attaches itself right onto that same C4b2a complex. This creates a new, bigger enzyme with a totally new job. And this new enzyme is the C5 convertase, C4b2a3b. See how that works? By just adding that one C3b component, the enzyme's function completely changes. It doesn't care about C3 anymore. Its new and only target is now the C5 protein. This brings us to the grand finale, the formation of the membrane attack complex, or MAC. All the steps before this, they were just the setup. This is the terminal phase, where the system delivers the final lethal blow to the target cell. So the C5 convertase does its job. It cleaves C5 into 2 parts.
C5a is a potent chemoattractant, which is like a flare signal calling in other immune cells to the site of infection. But the star of the show right now is C5b. This fragment is the seed that starts the assembly of the ultimate weapon.
So C5b is super unstable. It immediately has to grab onto a C6 protein just to stabilize itself. This C5b6 complex then recruits C7, which acts like a hook, letting the whole structure insert itself into the pathogen's membrane. And finally, C8 joins the party, drilling even deeper into the membrane and anchoring the whole complex right where it needs to be. And here's the really cool part. The C5b8 structure isn't the weapon itself. It's the scaffold for the weapon. It acts as a catalyst, recruiting a whole bunch of C9 molecules, up to 16 of them. These C9s then insert into the membrane and assemble themselves into a ring. And that ring of C9 molecules, that is the final membrane attack complex, C5b9. It's a hollow transmembrane channel. It's basically a giant pore that gets punched right through the cell's defenses. This pore totally destroys the cell's osmotic balance. Water rushes in, and the cell swells up and just bursts. A perfect kill. You know, a system this powerful is kind of a double-edged sword. If it went rogue, it could easily start attacking our own cells. That's why the body has built-in safety protocols. Regulation isn't just important, it is absolutely essential to prevent catastrophic self-damage. Failures here lead to very serious diseases. So the primary off switch is the C1 inhibitor. This protein acts right at the very beginning by binding to C1R and C1s and basically pulling them out of the C1 complex. This shuts down the entire cascade before it can even get started. A deficiency here, by the way, is what causes hereditary angioedema. Another key protector on our own cells is called decay accelerating factor, or DAF. If a C3 convertase accidentally assembles on one of our host cells, DAF acts instantly to just break it apart, dismantling that amplification engine. And the absence of DAF on red blood cells, well, that's the cause of paroxysmal nocturnal hemoglobinuria. So from that initial antibody targeting all the way to the final MAC attack, the classical pathway is just a masterpiece of immune precision. If you found this explanation helpful, then please like, comment, and subscribe.